I'm Joe Graydon.
And I'm Terry Graydon. Welcome to this podcast of the People's Pharmacy.
You can find previous podcasts and more information on a range of health topics at peoplespharmacy.com. Immunotherapy has become the latest innovation for treating cancer. Why did it take more than 100 years for this therapy to catch on with oncologists? This is the People's Pharmacy with Terry and Joe Graydon.
At the turn of the 20th century, a young cancer doctor in New York City named William Coley discovered that cancer patients with a nasty infection and a high fever sometimes saw their tumors shrink and disappear. He turned his discovery into a surprisingly successful treatment.
Have we made any progress since Dr. Coley introduced his toxin to treat sarcoma? How important is a fever to mobilizing the immune system against cancer?
Coming up on the People's Pharmacy, we'll talk about the reinvention of Coley's toxins, the first immunotherapy for cancer.
First, the news.
In the People's Pharmacy health headlines, a new study published in JAMA suggests that aggressive blood pressure control may have modest benefit for cognitive function. In 2010, researchers recruited 9,300 people over 50 to participate in the SPRINT trial, that stands for Systolic Blood Pressure Intervention Trial. The participants all had high blood pressure and were randomly assigned to get their blood pressure down to 120 or under 140. People in the intensive blood pressure control arm were slightly less likely to develop dementia during the 5 years of the study, but the difference was too small to be statistically significant. Mild cognitive impairment occurred in 14.6 cases per 1,000 person years in the intensive treatment arm. People in the standard treatment arm had a rate of mild cognitive impairment of 18.3 per 1,000 person years. This difference was statistically significant. The authors concluded that among adults with hypertension, intensive blood pressure control did not significantly reduce the risk of probable dementia.
A year-long investigation by Bloomberg News has revealed substantial concerns about the quality of generic drugs. According to the report, 90% of the medicines Americans swallow are now generic. The FDA does little, if any, testing itself, relying largely upon data from the manufacturers. This means that they trust generic drug companies to submit accurate data. Bloomberg cites the results from an online pharmacy startup called Valisure. The company tests medications that it dispenses. In the last year, it found that about 10% of the drugs in its inventory didn't dissolve as expected or have the labeled amount of active ingredient. While Bloomberg documented problems with some U.S. manufacturers, the expose also targeted the problems with drugs made in China and India. There have been numerous recalls over the last several months of blood pressure medicines, including valsartan, losartan, and irbesartan. An FDA inspection of the Zhejiang Huahai Pharmaceutical Company in 2017 found serious quality problems, but allowed the company to continue producing generic drugs. In July of 2018, the recall started with contaminated blood pressure medicine produced by this company.
Over the last 6 months, the FDA has announced numerous recalls of blood pressure medicines. Dozens of batches have been pulled from the market due to contamination with potential carcinogens. The FDA doesn't count the number of pills involved, but we suspect the number could be in the hundreds of thousands or even millions. As a result of the recalls, FDA has just announced a shortage of valsartan. Related drugs such as losartan and irbesartan are likely to be affected in the future. The FDA maintains that the overall risk is low. However, people who can't get supplies of their usual prescription blood pressure medicine may need to ask their health professionals about alternatives.
Elevated LDL cholesterol is widely recognized as an important risk factor for coronary heart disease. A new genetic study involving more than 650,000 participants over roughly 7 decades demonstrates that high triglycerides are equally important. The study concluded that lowering triglycerides could be just as beneficial as lowering LDL cholesterol. Eating fewer processed foods and simple carbohydrates is an effective strategy for lowering triglycerides.
People in chronic pain often feel abandoned by the medical profession, especially as physicians are cutting back on prescription pain relievers such as opioids. A new study published in the journal Evidence-Based Mental Health suggests that mindfulness meditation can help people cope much better with their pain. The researchers reviewed trials of cognitive behavior therapy or mindfulness-based stress reduction for pain management. They found 21 trials of almost 2,000 people were appropriate for analysis. Although only one trial compared these two approaches directly, the investigators found that each technique had comparable effects on reducing pain intensity and psychological distress. And that's the health news from the People's Pharmacy this week.
Welcome to the People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. Our topic today is immunotherapy to treat cancer. This may seem like the absolute latest innovation in oncology. However, it has roots going back more than a century. What was the first immunotherapy for cancer? And why was it overlooked for so much of the 20th century?
To help us understand this fascinating history, we turn to Don McAdam. He's the former head of MB-Vax Bioscience and the author of The Reinvention of Coley's Toxins.
Welcome back to the People's Pharmacy, Don McAdam.
Nice to be back with you.
Don, immunotherapy, it's like the most exciting advance in cancer treatment in the 21st century. We've got checkpoint inhibitors like Keytruda and Opdivo. They run anywhere from $100,000 to $150,000 a year for treatment. Then there's CAR T. It can cost anywhere from $350,000 to $450,000 for a course of treatment. But even though this is the latest advance against cancer, the use of the immune system to fight cancer goes back over 100 years. Can you please tell us about William Coley?
Well, William Coley was the first physician to cure any disease with an immune therapy. And that disease happened to be cancer, and it happened in 1891, which was the 19th century, not the 21st century.
Tell us a bit more. How did he do this, and what made him think of it?
Well, the first patient that Coley treated for cancer when he was 27 years old was a young lady called Bessie Dashiell. She happened to be John D. Rockefeller Jr.'s girlfriend, and she developed a sarcoma in her wrist. So Coley provided the standard therapy for a sarcoma, which was amputation of the limb, thereby removing all of the cancer. But the cancer returned, and in a couple of months, she was dead. It was a terrible impact on Coley because this was his first patient. So he immediately set out to find how he could treat the next sarcoma patient that happened to come his way. And the story about that is a little bit more complicated. In 1884, there was a patient who presented himself to the New York hospital, and he had a deep-seated sarcoma in his neck. This was a patient called Klein. And Mr. Klein had a series of surgeries, and none of them were successful. The final surgery in 1884 was complicated by a concurrent infection. That is, after the surgery, the patient was infected with a bacteria called Streptococcus pyogenes. And that bacteria caused a disease called St. Anthony's fire, which is a disease in which the head turns a blistery scarlet red. So it's a very attractive disease, but it's also a potentially fatal disease. Nevertheless, the disease ran its course in this particular patient, and when the disease had run its course, the cancer was gone as well. Now, this patient was discharged from the hospital 7 years previously and had not been heard from since. So Dr. Coley went off to the Lower East Side of Manhattan, where the German immigrants at that time were living, and found Mr. Stein. He was still alive, brought him back to the hospital. They checked him out. The cancer was completely gone. So Coley reasoned that this infection caused the cancer to completely regress, and he decided to treat the next appropriate patient with the same kind of cancer by infecting that patient deliberately with this Streptococcus pyogenes in order to induce a regression.
Now, when Dr. William Coley discovered this patient, it was like the light bulb goes off in his head. But he also starts doing some research, and he discovers that German physicians had actually been writing about this, we'll call it pre-immunotherapy, decades ahead of that.
Yes, that's true. There were two Germans, a Busch and a Felisen, and these two physicians in Germany had done some experiments with patients who had cancers by wrapping them in the bedclothes of a previous patient who had died of scarlet fever, for example. And they wrote about the results, and in some cases, they achieved some temporary remissions of disease. But neither Felisen nor Busch ever turned this into a therapy.
So what did Dr. Coley do once he had this light bulb phenomenon of, hey, infection, fever, may actually have some benefit against cancer?
Well, what he did was the next appropriate patient was a young man with a deep-seated inoperable tumor in his neck, much like Mr. Stein had had. And so Coley injected him with live bacteria. It took a while to actually inject him successfully and develop the infection. But eventually, Coley produced an infection that induced an immune response that caused the cancer to completely regress. And the patient became cancer-free and returned to his home in Italy and apparently lived another 7 years and died of some other condition.
That must have been pretty amazing.
Yes, it really was amazing. In fact, Coley treated in total 12 advanced cancer patients with live bacteria, and there were complete responses. 2 of the patients had complete remissions, meaning that there was a complete absence of cancer, but the other 10 patients did die. This was not unexpected because these were terminal cancer patients. What was unexpected is these 2 patients that died did not die of cancer, but they died of the infection that Coley caused.
Dom, it's pretty much frowned upon to give your patients a therapy that's going to kill them. So I'm sure that Coley was horrified when his patients died. What did he do then?
Well, what he did is he decided to try to treat patients with killed bacteria rather than live bacteria. So he came up with a intuitive formulation of 2 types of bacteria that represented the whole range of bacteria available in the world, called gram-negative and gram-positive. He made sure one of them was the bacteria that caused the disease that led to the previous regression. And the first patient to receive this new product, which is now called Coley's toxins, was a 16-year-old boy who was bedridden with sarcoma. And this patient had a deep-seated tumor that was 5 inches on a side, like a big cube of cancer, that was inoperable because it was attached to the underlying bones. So over a period of a number of months, Coley injected his toxins every few days directly into the tumor, and it gradually reduced in size. The cancer completely regressed. The boy returned to his regular work, and he remained in very good health until he died of a heart attack 26 years later.
Don, when Dr. William Coley had this, what could only be described as amazing success with a handful of patients, and began to realize that creating this toxin instead of the live bacteria, that he was really onto something. And I'm guessing that he treated a whole lot of patients over the course of his career. How did he do?
Well, he treated about 1,000 cancer patients, but more importantly, before the turn of the 20th century, at least 42 other physicians from Europe and North America had reported cases of cancer they had successfully treated with Coley's toxins. And the reason they were able to do that is by the turn of the 20th century, both Park Davis in the United States and the Lister Institute in the United Kingdom were manufacturing Coley's toxins and distributing them worldwide.
How long did that go on?
Well, that went on until the rise of chemotherapies in the 1950s. At that time, the middle of the 1950s, the Park Davis company in the United States ceased productions of Coley's toxins. And that might have been the end of the story of Coley's toxins because, there was now this new thing called chemotherapy, which was so much better.
How much better was it?
Well, initially, it was hard to tell. With any new cancer therapy, you really can't judge it until some time later, because what's important is not the ability to induce a remission, which is what we look for today. You get chemotherapy, it induces a remission, and you live longer. What's really important is to induce a regression where the cancer begins to be destroyed, and in fact, the cancer is cured, and that cure persists for a long period of time. That's called in the trade a durable, complete response. And that's what really is needed. And what happened is by the 1960s, there was a resurgence of interest because it became clear to a small but dedicated group of researchers that Dr. Coley's historical results were much better than would be expected for a similar group of contemporary patients. And the reason was Dr. Coley's patients, a good number of them, achieved durable responses. As an example, in 2009, two leading researchers at the MD Anderson Cancer Center in Houston wrote a paper in which they revisited Coley's legacy. And they wrote, and this is a quote, that Dr. Coley achieved long-term cure rates unrivaled by medical science in the 73 years since his death. That's amazing. What they're saying is not that he had better 5-year survivals, not that he had better remissions or whatever. What he did do was he cured more of the cancers than are cured today. Because the problem we have with our existing chemotherapy and radiation is that while that can sometimes be a good response if the tumor has not metastasized, has not spread, if there's been a spread of cancer to the other organs, chemo and radiation isn't very effective because the chemo and radiation harms the immune system as well as harming the cancer. So when the cancer returns due to a metastases, the patient has no natural defenses to it.
Now, Don, in the early days, Coley's work was actually rejected by many of his medical colleagues because in those days, surgery was the number one, in fact, the only way to treat cancer. And when Coley reported remissions, in fact, cures, they said, oh, well, it must have been a misdiagnosis. They couldn't have really had cancer after all.
Yes, that was a major problem for Coley in the early days, but he really didn't know he was getting cured. You see, that's the issue. It wasn't until after Coley died in 1936 that his daughter, Helen Coley Nautz, who originally intended to write his life story and just organize his papers, became intrigued with all of these cases and began following up and became an autodidactical, self-learned immunologist and established the Cancer Research Institute. She recruited Lloyd Old to help her with that. And the two of them eventually rehabilitated Coley's name and established by careful documentation that the patients he did treat and the patients that other people treated who did survive their diseases had long-term cures of their diseases, which is something we don't see in chemotherapy and radiation.
Don, we now live in the 21st century, and immunotherapy is, in fact, the cutting edge of cancer treatment for many kinds of malignancies. How does Coley's toxin compare in either effectiveness or safety to modern-day immunotherapies like CAR-T or perhaps those checkpoint inhibitors like Keytruda and Opdivo?
Well, unfortunately, I think most of the modern immunotherapies that are being sold for like pharmaceutical companies have some efficacy, but they're grossly overpriced, and they're taking advantage of cancer patients. And they're being sold on the basis of you might live longer. They're not really attempting to cure the disease. They're just attempting to sell products. I hate to say it, but it's the truth. The way to treat cancer is with targeted therapies. For example, antibodies would be fine. We had a patient who was getting NY-ESO-1 antibodies through Lloyd Old at Memorial Sloan Kettering. They combined that therapy with Coley's and with a number of other therapies. And this particular patient, Renee Che, who I talked about in the book, had a terminal form of cancer called synovial sarcoma, and she was completely cured of that disease, even though in the course of her therapy she had to do horrible treatments to her body, including the removal of part of her jawbone. But she's now been cancer-free for 7 months. It's unheard of. But I think the future of cancer immunotherapy is some form of combination therapy. And one portion of that therapy is something that can induce and accelerate the immune response. And at the moment, Coley's is the best thing that we have to do that.
Don, Coley's toxin has been available for over 100 years, but it kind of fell into disfavor in the last half century. And for the most part, modern-day cancer researchers have been looking for other ways to stimulate the immune system. Do you see the possibility of Coley's toxin being resurrected?
Absolutely. I think Coley's toxin, in combination with some of these other immune therapies, hopefully inexpensive immune therapies, will be a real alternative to chemotherapy and radiation for the treatment of cancer. And just to address that point about Coley's falling into disrepute recently, in fact, in that paper that Decker and Safdar from MD Anderson wrote, where they pointed out that Coley achieved long-term cure rates better than would be expected today, in two forms of cancer, breast and lymphoma, Coley's cure rate as a single agent was 14%. The MBVax cure rate was 32%. In lymphoma, Coley's cure rate was 38%. The MBVax cure rate was 44%. So I think the version of Coley toxins that we manufactured, we called it Coley fluid, was in fact better than the version that was manufactured for the personal use of Dr. Coley. And in the book are the complete manufacturing protocols. In fact, the reason why I wrote the book was to make the complete manufacturing protocols and how the vaccine was designed, make that available to anybody who wants to use it anywhere in the world, companies or individuals, free and at no charge for all time.
So are there clinical trials going on now using that approach?
No, there are no clinical trials going on. There is no vaccine available right now except from a small lab in New Zealand, and hopefully that lab will reformulate to make the same vaccine that we used to make, and at least we'll have that to count on.
Don, modern immunotherapy is unbelievably expensive. As I mentioned earlier, it can run $100,000, $150,000 for a course of treatment. The new CAR T immunotherapy can run $350,000 to $450,000, and there's even talk of a new immunotherapy that may cost as much as $1 million. How much is Coley's toxin for a course of treatment?
That's completely absurd. It's true, but it's just completely absurd. Coley toxins is virtually, the cost of the ingredients are practically nothing. The cost of the facility is very small. I put together a business model for the laboratory in New Zealand where they can make a profit selling the vaccine. We never sold it. We gave it away for free, but they could make a profit selling the vaccine for $150 a vial, one vial being enough for approximately 30 intravenous infusions, which works out to $5 per therapy. I should point out that there are pharmaceutical companies in the U.S. X-Biotech comes to mind. I happen to be a director, so I have a vested interest. But X-Biotech is manufacturing human antibodies at much, much lower cost to manufacture than any of the pharmaceutical companies by having entirely new technology. And they've been able to harvest antibodies directly from human immune systems, so the antibodies are not immunogenic and produce zero side effects. And I think those kinds of companies should be the future of immune therapies, and the larger pharmaceutical companies will just have to get on board and start pricing their products at an appropriate price.
We've been talking about using Coley's toxin to treat cancer, and we actually haven't even been very specific about the types of cancers that might be treated. Apparently, Dr. Coley treated a lot of sarcoma. He may have treated some other types as well. Are there other diseases besides cancer that might benefit from this kind of immune therapy?
Yes, there are. Let me just very quickly say that at MBVax, patients achieved complete regressions who had inoperable or metastatic breast cancer, lymphoma, melanoma, lung cancer, esophageal cancer, and stomach cancer. And before we were shut down by the FDA and Health Canada, we supplied coleus to veterinarians to treat 3 dogs with terminal cancer, 2 of which completely recovered. In the historical record, fever therapies such as coleus, toxins, and others were used to treat a wide range of non-malignant disease. The best documented non-malignant condition is arthritis, where physicians in the 1920s treated arthritic patients and achieved cure rates for arthritis of more than 50%, and published 2 papers in the 1920s that detailed their cures and their results. And I think the fact that arthritis is still considered an incurable immune therapy when it was routinely cured almost 100 years ago is a medical disgrace. Also, diseases like multiple sclerosis has been treated in the past with fever therapies, and Crohn's disease or ulcerative colitis has been treated in the past with fever therapies. And there's a whole literature on diseases that have been cured or treated successfully to reduce symptoms using fever therapies, which my partner Stephen Hopchin-Can at the University of British Columbia knows more about than anybody in the world, and one day he'll probably write about that.
So, Don, in summing up the reinvention of coleus toxin and the reason that you wrote this book, what would you like to see evolve? What would you like to see for the future of cancer treatment?
I would like to see manufacturers of coleus toxins make the product according to my formula. It's free to make. There's no intellectual property you have to buy. There are many, many clinics, not in North America, however, that can freely use it today. Clinics in Germany, for example, clinics in Australia and New Zealand are allowed to treat terminal cancer patients because terminal cancer patients are allowed under their law access to unapproved medications and it's only right if that's the case. I think any cancer patient who's considered terminal should be allowed to use unapproved medications, providing a physician is prepared to treat them. I certainly do not approve of quack therapies. We don't want to get people producing products that don't work. But if products do work and a physician is willing to treat a patient with something that they believe is good for that patient, and that patient is considered terminal, I think it's the responsibility of the government to allow those patients access to these therapies.
Are there any places in the world where patients can access coleus toxins?
Well, many places. My sister, for example, was diagnosed with metastatic breast cancer in 2011, and I sent her to Dr. Decio Basso's clinic in Paraguay, where she was treated for 3 months. His treatment of her resolved the metastases. When she came back to Boca Raton, Florida, where she lived, she traveled back and forth to the Bahamas for some additional therapy with Kevin Bethel, a physician there. And then eventually she had a small amount of chemo to debulk the remaining tumor, and then the rest was removed surgically. So today she's been cancer-free now for about 8 years, something like that, and she's happy and healthy. And the point is, in her case at least, coleus toxins did not cure her, but coleus toxins did destroy the metastases that probably would have killed her.
Don McAdam, thank you so much for talking with us on the People's Pharmacy today.
Thank you.
The People's Pharmacy podcast is sponsored in part by Kaya Biotics. KAYA Biotics offers the first probiotics which are both certified organic and hypoallergenic.
All probiotics are produced in Germany under laboratory conditions with high-quality ingredients and under strict regulatory oversight.
The 3 available formulas are created for very specific purposes, such as strengthening the immune system, fighting yeast infections, and helping with weight loss.
To learn more about Kaya Biotics probiotics and the important topic of gut health, you can visit their website, KayaBiotics.com. That's Kaya Biotics.com. Use the discount code PEOPLE for $10 off your first purchase.
Welcome back to the People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. If you would like to purchase a CD of this show, you can call 800-732-2334. This is show number 1151. The number to call, 800-732-2334, or you can place the order online at peoplespharmacy.com. If you'd like, you can also download the podcast from iTunes.
The People's Pharmacy is brought to you in part by Kaya Biotics, probiotic products made in Germany from certified organic ingredients. That's Kaya Biotics.com.
Today, we're talking about the first immunotherapy for cancer and how it has been followed by modern-day oncology treatments. Coley's toxin is almost forgotten, but the 21st century has seen approval of new immunotherapy treatments.
We turn now to Dr. Sharon Evans, professor of oncology in the immunology department at Roswell Park Comprehensive Cancer Center in Buffalo, New York. Dr. Evans is a past president of the International Society for Thermal Medicine, which earlier this year honored her with a J. Eugene Robinson Award. Her research program is focused on the inflammatory cues that positively or negatively impact our protective immune system during infection or medical treatment, such as cancer immunotherapy.
Welcome back to the People's Pharmacy. Dr. Sharon Evans.
It's nice to be with you.
Dr. Evans, it seems like the hottest area of research today in cancer is your field, cancer immunotherapy. And I think back more than 100 years to Dr. William Coley, who in a sense was the father of immunotherapy for cancer. But sadly, his research was, kind of disregarded. It was dismissed for decades and ultimately fell into disfavor. Now it's back, not so much his research exactly, but immunotherapy. It costs an arm and a leg, $100,000, $150,000. Some kinds of immunotherapy, $500,000 or more. The response rate is highly variable. Some people are almost cured, durable remissions. Other people, not so much. Some people get really seriously ill, adverse reactions that are just horrific. And I'm just wondering, have we made a whole lot of progress since Dr. Coley's days? I mean, his response rate was pretty good.
You bring up a question that really resonates right now because there's so much excitement. Let's face it, we had the Nobel Prize in Medicine and Physiology going for immunotherapy, which I might mention is the very first Nobel Prize that has been given for the treatment of cancer. There have been prizes given for understanding how cancer grows and the biology of cancer, but not for the treatment. So it really stands out how people are excited. And I think it's fair to say that we have progressed tremendously. The issue about when Coley was doing his research, which involved live bacteria, very inconsistent preparations of bacteria that were given to patients to try to boost their immune response. The problem is the science wasn't there yet, so there was no understanding of how it might work, why some patients responded and some didn't. So it fell out of favor because of the inconsistency. We have inconsistency now in immunotherapy, as you raised, but we understand much better about the mechanisms. And as we piece away at that, chip away at understanding that, there's that real opportunity to increase the number of people that can respond. Because the people that do respond tremendously. Literally, the tumors melt away, and they are protected long term. So that's why there's so much excitement.
Dr. Evans, I wonder if you can explain to us some of those mechanisms that we now understand or think we understand.
So I think it's all, our understanding is really on that framework that we now get a better idea of the various complex pieces of the immune system. And our response to cancer is no different than when we get a cold. It's that idea that you can ramp up your immune system to quickly recognize that you're under attack, whether it's influenza or whether it's cancer, and then mount a counterattack. And so there are various cells in our body that we all know as part of our white blood cell components that come together in a highly controlled and integrated way, because the good side is the immune system can protect you. The bad side, if it's not controlled, then you have very strong immune responses and side effects and so on. So you're almost on a teeter-totter in terms of appropriate balance for whether you get a good immune response to cancer or, in fact, the cancer wins.
You know, the traditional treatments for cancer for many decades after Coley was surgery, it was radiation therapy, it was chemotherapy, really toxic chemicals. And the whole idea was, well, maybe we can get the entire cancer and remove it surgically. And if we can't do that, then we'll poison it with chemistry and we'll try and kill every single last cancer cell, which I suspect we now know is almost impossible. Or we'll try to use radiation to kill all those cancer cells. Is it possible that some of those treatments that may be now considered somewhat old-fashioned for certain kinds of tumors may have been working through immunotherapy and we just didn't realize it?
In fact, that's exactly where the field has come. Over the past, I'd say 10 years, we've come to appreciate that these standard pillars that you talked about, particularly radiation and chemotherapy, they poke at the immune system. So the way the tumor cells are killed during these standard pillars of treatment approaches, chemotherapy or radiation, is activating the immune system. Again, very much in the same types of mechanisms that a flu would activate your immune system. So the type of cell death will prompt or poke or tell the immune system, in fact, there's a danger signal. But it so happens in cancer, it's almost a danger signal coming from within rather than an outside invader like a pathogen or a microbe. So it's called immunogenic cell death, this idea that you're activating the immune system and harnessing this anti-tumor effect. That being said, generally, these standard therapies that you're mentioning are not so strong at activating the immune response because the cancer's been fighting back. It's been blocking the immune system all along the way. So you generally need to have several layers of protection. So as an example, chemotherapy alone can bump up the immune system and get a better response, but it's not very long-lasting. You combine that with an immunotherapy to boost the immune response, and when you get the two together, you can have a tremendous effect. And that's really where we are in 2018. Moving into 2019 is this notion that you use combination therapies to combat the immune response at different angles to attack.
One of the things that was fascinating about Dr. Coley's approach was that he would inject his immunostimulant material, so-called Coley's toxin, directly into those sarcoma tumors. And sarcoma is one of those really nasty cancers. The response rate was generally not very good with surgery alone. And he tried to induce a fever. In other words, he sort of titrated the treatment. He gave these injections repeatedly over several weeks and months to get a fever maybe of 102, 104, maybe as much as 106. And the fever would last for a couple of hours and then it would gradually fade away. It was as if he was constantly trying to teach the immune system to go after that tumor. Are we beginning to try to do somewhat similar things today?
So some of the immunotherapies, you mentioned earlier that many of them have very toxic side effects. Usually what that's associated with is what we call a cytokine storm, a very inflammatory response to the therapy, which very, very often is accompanied by fever. So there are several pieces of that whole story that William Coley was pushing and that has been substantiated in retrospective studies. The patients that did the best were the ones that had the highest fevers and more prolonged. So this notion that the fever response could be helping, it's either a readout that you're having a good immune response, but also the fever response itself actually prompts the immune system. So it works together with the agent that's activating. So really there's sort of a synergistic, I would say, or a cumulative effect of having different types of responses. So it's not, I wouldn't say that people are documenting the level of fever when patients are treated with immunotherapy now as an endpoint or an outcome, but it's certainly appreciated that a lot of patients get these kinds of responses that may help promote the ability to be protective with immunotherapies.
It sounds like you're suggesting that the fever is much more than a marker of an immune response, but actually may be participating in some way in the immune response. Would that explain why occasionally we hear about people who have a spontaneous remission of their cancer after they have an infection that caused a high fever?
It's possible. Those kinds of anecdotal analyses are hard to do, but in fact, that's exactly how William Coley got into this whole idea of trying to generate a fever, is he went back through medical logs from all over the world and made a record and showed that patients that got spontaneous infections that were associated with high fevers, and not usually for hours, we're talking about prolonged fevers, 12 hours, 24 hours, weeks, they often had evidence of spontaneous rejection of tumors. So that was kind of where the idea really started in the first place. So many of us have worked on the febrile element. In other words, the idea that the temperature component of fever might be amping up the immune response, and have found some very profound effects, not only for infectious disease, but for fighting cancer. It acts at every level of the immune system, which is in fact exactly what immunotherapies do. They target every level of the immune protective system of our body.
Can you tell us just a little bit more about that, please?
Well, so in our own research here at Roswell Park, one of the things that we've been very interested in is how the temperatures that are above your normal body temperature, so as you said, Joe, anywhere from 99 degrees up to 105 degrees, how that changes where immune cells position themselves in the body. So it turns out that our immune cells are protective, but they don't just protect walking down the street. They have to be in the buildings to do their work. So they have to be in the tumor. That makes a lot of sense. But they also have to be in your immune organs, like your lymph nodes, that we all feel when we get a cold and they get swollen. So that's where the work is done. And it turns out that fever brings cells together so that they are more efficient at getting exactly into the tissues they need to be in. Just one example. It also lowers the threshold for cells to signal and respond and become active killing cells. It literally can be very effective in amplifying the ability of cells to kill as well. So it's really quite multifunctional in terms of that aspect, and not all that well appreciated.
Let's talk a little bit about that lymph node system. I've always been kind of fascinated that we have this circulatory system, the lymph system, that is really critical to the body, but it's not like blood vessels where we can document, arteries and veins. It's a little more amorphous. And it's not uncommon for surgeons to remove lymph nodes because that's where cancer cells can sometimes migrate. I'm wondering if we're starting to rethink removing lymph nodes and what role the lymph system plays in monitoring and even treating cancer.
So I think the jury is still out. Those of us that have focused on the lymph nodes that surround the tumor, and there can be many, and as you mentioned, at surgery, as many as 10 lymph nodes can be taken out because of the importance of staging. That's been somewhat controversial until several very elegant studies were published this summer in Science that showed that, in fact, tumor cells can go through those very lymphatics that you talked about, drain down to the lymph nodes, and then use the lymph nodes as their exit strategy, their doorway to get throughout the blood to the rest of the body and metastasize. So in that case, I think the idea that the tumor cells are there, it would make a lot of sense to remove them. The counterpart is that we all know that the immune system is best in activating a response that we need in immunotherapies in the lymph nodes. So it's going to be tricky. I think as humans, we have some advantages. We have over 450 lymph nodes in our body. So we take out a few, we have a lot of backups. So I really think there's going to need a lot more very systematic comparative type research to figure that out. A lot will depend on what stage a patient is at, if they're early stage versus late, I think, to answer that very question.
Dr. Evans, we have focused for years on the cancer. You know, what kind of mutations, how aggressive the cancer, stage 1, stage 3, 4. But there's been a growing interest in what's called the terrain, the surrounding environment, the body itself, the tissue, where the tumor is, and how the tissue is responding to that invader, which is actually, after all, us. Can you talk a little bit about the terrain?
Absolutely. I think you've hit on the golden nugget of where our understanding has advanced so much just in the last 10 years relates to the terrain. You take cancer, and we used to all think of it as a very consistent blanket of a mass of tumor cells, and the rest didn't matter, except maybe to hold it all together and give it a little structure. Now we realize that a large part of the cells in that terrain actually are our own body's immune system. So while the basis of immunotherapies are to drive a reaction against the tumor to kill it and reduce it, shrink it, literally shrink it. But there's a flip side, and that's where that teeter-totter of the immune system comes in. The flip side is our immune system is geared to prevent too much reaction going on. And so a lot of the terrain is made up of various types of white blood cells that block the immune response. And so not only do they block the immune response, so there isn't a good killing effect and reduction of tumor size or volume, but on top of it, they actually actively cause the tumors to grow. And they replenish that terrain with more and more blood cells, which bring more nutrients, so that cancer has an advantage. So that's where I think the excitement is that we now understand it's a very, very complex map of what's going on in the tumor microenvironment. There are active therapies that are being combined with immunotherapy now to try to eliminate some of this terrain effects so that the immune system has a better chance.
You're listening to Dr. Sharon Evans, Professor of Oncology in the Immunology Department at Roswell Park Comprehensive Cancer Center in Buffalo, New York. Dr. Evans is a past president of the International Society for Thermal Medicine. Her research program focuses on the inflammatory cues that positively or negatively impact our protective immune system during infection or medical treatments.
After the break, we'll find out how active therapies to kill cancer cells are used in conjunction with immunotherapies.
You're listening to The People's Pharmacy with Joe and Terry Graydon.
If you value the health information you get when you listen to The People's Pharmacy, consider subscribing to our email newsletter. You'll get the latest health news and information on upcoming podcasts delivered to your inbox twice a week. Look for the link at peoplespharmacy.com. Welcome back to the People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. If you'd like to purchase a CD of today's show or any other People's Pharmacy broadcast, you can call 800-732-2334. Today's show is number 1151. The number 800-732-2334, or you can place the order online at peoplespharmacy.com. You can also download the free podcast from iTunes or from our web store. We invite you to consider writing a review.
The People's Pharmacy is brought to you in part by Kaya Biotics, probiotic products made in Germany from hypoallergenic organic ingredients. That's KAYA Biotics.com.
Commercials on television promote the idea that immunotherapy drugs like Keytruda and Opdivo can prolong life for cancer patients. What should you know about immunotherapy and other innovative cancer treatments?
We're talking with Dr. Sharon Evans, professor of oncology in the immunology department at Roswell Park Comprehensive Cancer Center in Buffalo, New York. Dr. Evans is a past president of the International Society for Thermal Medicine, which earlier this year honored her with the J. Eugene Robinson Award. Her research program is focused on the inflammatory cues that positively or negatively affect our protective immune system during infection or medical treatments such as cancer immunotherapy. We were just talking about combination therapies.
What are those active therapies that are being used in combination with immune therapies?
So some of them involve immune boosting type therapies where components are used that are actually almost replicating an infection. So bacteria, either in pieces of bacteria or killed bacteria or inactivated bacteria, is one area where people have been, over the past decade or so, adding that in combination because that will tip the teeter-totter towards an immune response that's more productive. Another are these types of white blood cells that are very, very potent suppressors. And so some standard chemotherapies like capecitabine, for example, which is a very standard of care chemotherapy, it can attack these immune systems that put a block on the ability of the other side of the immune system to attack. So a lot of these therapies, where immunotherapy treatment is now, it's not only being used as a single agent, depending on the disease, but also in combination with various agents that will ramp it up in its favor. So those are just a couple of areas, really trying to strategically target the immune system that is blocking.
Dr. Evans, you bring up a fascinating concept, and that is multiple approaches. Immunotherapy, where there might be what's called a checkpoint inhibitor, there might be a traditional chemotherapy drug, there may be something entirely different, all used together in order to go after hard-to-treat cancers. How can oncologists keep up with the advances that seem to be happening on a monthly basis, especially when it's these novel approaches that you're talking about? And then, more importantly, when we discover something that's a real advance, how do we get the FDA to approve them and insurance companies to pay for them? Because if you have to pay out of pocket, it can be hundreds of thousands of dollars a year.
For certain. I think the reality, and we're always reminded of that in research. In research, we can combine multiple modalities, and the cost is not, or the consequence is not a major determinant. But then when we speak with our colleagues, who are the clinicians who are actively at the front treating the patients, it's just, managing the reality. Also, with more and more treatments layered on top, as effective as they can be, a lot of the cost comes from the fact that the patients have to be monitored so carefully. They may have to be in hospital and so on. So, there are a lot of hidden costs besides the cost of the reagents. So it is challenging. I can tell you that I monitor clinical trials in certain areas like breast cancer every week. When I'm giving seminars, I may have to change my slide from yesterday to today to just make sure it's up to date. So it's moving so quickly that I don't think it's realistic that a general clinician would be able to keep up. Where they depend on are the major trials and the things that move to the front. So as you know, FDA approval depends on showing safety and efficacy. So unlike the clinical trials where that is sorted out, the general oncologist has the advantage of having major trials with thousands of patients, and then the cream goes to the top in terms of which are the most promising. So, I think that's the only way it really can be managed at this point. Certainly the specialty institutes are the ones that are kind of pushing this forward because they are trying these compounds and the various combinations before it's approved and generally commercially available.
Do we have any way of telling ahead of time which patients might respond best to a given therapy or a combination therapy?
So, Terry, you bring up exactly what we call the holy grail in terms of immunotherapy. That is the biggest challenge because, as Joe, you brought up early on, some patients respond remarkably well, and that's why there's so much excitement. And yet others, disappointingly, don't respond at all, or they start and they respond, and then they have what we call acquired resistance. Several months, even years down the line, they become resistant to the therapy. So there's a major push to try to identify what we call biomarkers, a readout that would tell you that this patient is likely to respond, this patient may not respond or would benefit from another type of therapy first and then come in with immunotherapy. This patient's never likely to respond. We don't have all those tools in place yet. The closest we are is that if you take out a biopsy, and reading the complexion of the tumor and the terrain that we've talked about can give some indications, but it's definitely not perfect. If you have a lot of immune cells locally at the tumor, that's a better chance for response, not only to immunotherapies, but chemotherapy and radiation as well. If certain cell types are there, you have a better chance of a response if they're immune cells, and if other, let's call them bad guy immune cells are there, or at least in the complexion of the tumor, they're not advantageous, then that's a possibility of a patient being resistant. But the problem we have is as these therapies work and the tumors shrink, we have less and less tumor to deal with to really get an idea of the status of a patient. So where everybody's really actively looking is trying to find a circulating marker that you could pull out of the blood. You could sample from a patient easily and readily and make a determination. And we're really, I would say, tip of the iceberg in trying to figure that out. Many of us are working on candidates and our favorites that relate to a mechanism, but there's a far way to go on that.
Dr. Evans, one of the great challenges for patients and their families has to do with what I'll call the old surgical approach. And what I mean by that is a surgeon removes, let's just say, a prostate gland and says, we got it all. And you have a low risk, a Gleason 6 or a Gleason 7, not so serious, and we got it. The lymph nodes were clear, the margins were clear. Or a surgeon removes a lump in a woman's breast and says, we got it. And then 2 years later, all of a sudden, something starts to show up again. The idea of getting it as if it was like one solid mass or, one cell, it's actually hundreds of millions of cells or maybe even billions of cells. It seems a little old-fashioned, which is why we want to be able to get those rogue cells that somehow escape surgery, or we didn't even know that they were gone a long time ago. We were told by Dr. Susan Love, a breast cancer surgeon and specialist, she said, by the time we see breast cancer on a mammogram, it's been growing for years. There is no such thing as early detection. There's just earlier detection. What would you say to your surgical colleagues who like to tell patients, we got it all?
Well, I think that most of the surgeons that I associate with are always very cautiously optimistic. Certainly the prognosis is better when you think you have a clean margin removed, but most surgeons are actually incredibly aware and humble of the fact that the risks are still there. Because, as you said, one of the things that has really tracked with how a patient can do are looking at these tumor cells that we call them circulating tumor cells. They're in the blood. They're moving around. And so you've taken out the main nodule in the prostate, for example, or in the breast. That doesn't get rid of what's been moving around and can actually lodge little nuggets in your tissues for very long periods of time. So generally, I think that most surgeons are very careful about how they phrase it. That being said, having been on the flip side, as we all have family members that have been faced with these kinds of situations. What you hear versus what's being said and how you interpret it can be not always matched. So I think that's where patient advocates and physicians' assistants and so many other people that are involved in trying to make sure the picture is clear are very, very important.
With the rapid advances that you all are making in your field, do you have recommendations for patients or their families on how to stay up with what might be the most appropriate treatments for them?
It's such a good question. We're facing a situation in the field that is unprecedented because normally the cutting-edge therapies that were available were relayed the information to the oncologists and the specialists, not to the public. And, I think we all see that. I mean, here we're talking about it right now on this show, that now the public is incredibly well informed, not only through the internet, but every time you turn on the news, you'll see immunotherapy discussed in a really big way, but in a way that patients don't really exactly know the pieces. So I often give talks to patients and very, very educated patients who are going through all kinds of treatment decisions. And I'll ask them, do you know what people mean when they say on commercials, TV commercials, my PD-L1 status allowed me to go on immunotherapy? And you'll have a room of 300 people, and maybe 2 people raise their hand. So I think the intent right now is to provoke people to want to get more information, read about it at a certain level, but actually ask the questions they need to ask of their doctors. That seems to be where we are in this whole field of more informed but not fully educated because the information isn't being given in that way yet. So I think the best is talking to specialists, truthfully, that deal with this day in and day out. This is their only treatment, that the only kind of disease that they're treating. So they'll be the ones that are very, very up to date. And then having a dialogue with those physicians, I think, is incredibly valuable in making decisions.
Dr. Evans, there is a website, I believe it's clinicaltrials.gov, where patients can actually find out who's doing what. And because patients are becoming so well educated about the latest and greatest treatments, especially in the field of immunotherapy, they can find out who's enrolling and if they can sign up and if they fit the criteria. Is that something people should be aware of?
Absolutely. Absolutely. And in fact, some of those, the clinicaltrials.gov that you're talking about is supported by the National Institutes of Health, but there are also subgroups that will be very disease-specific. So they'll list only the trials in breast cancer, only the trials in melanoma. So it's a great starting place. The thing I would add, though, is that when you go into those, and they're not really intended for the general public in the sense of ease of use. So you have to click into several tabs to get to where the institutes are that are running the trials. But if there's something of interest and a local institute isn't listed, it's still worth asking because those trials are, that website is not, even though it's so powerful, because the trials are going online so quickly, it's not always up to date of all the institutes that are involved. So there will be, I know exactly of examples where not all the institutes will be listed, so it's worth finding out, and you'll find out that there will be someone, a local institute that's running the trials.
Dr. Evans, when you look into your crystal ball, when you look at the research that I am sure you review in the form of grants going to the National Institutes of Health, what do you see? What is your hope for the future? Where is the optimism?
So, of course, I'm pretty biased in my answer, being an immunologist and a long-term immunologist with an interest in cancer. I think the idea that we may be able to harness this very, very durable response is absolutely where the enthusiasm is. You know, we've been talking about William Coley's work. One of the things that was the bellmark of that work is that while they injected the agent into the tumor to try to kill the tumor, often they saw effects far away in tumors that were, say, you inject in the arm, you would get protection in the leg. That happens in radiation as well. So that idea, the only way that can happen is through the immune system, which is absolutely elegantly equipped to get to different parts of your body. So I think the promise is so strong. The more we understand the microenvironment, or as we call it, the tumor microenvironment, the terrain, and how we can play with that balance. The biggest thing I think is that where we've been focusing the last 10, 20 years is what's going on inside the tumor, even the immune complexion in the tumor, where I think the untapped potential is to recruit the whole body, because that's how the immune system really works. So if you can be replenishing the killer cells in the tumor continuously by using your whole body's immune system, that's where it's going to be really profound. There are roadblocks to it. The Nobel Prize-winning immunotherapies that we're also aware of is one piece of that puzzle. I think chipping away at what the other opportunities, it will be profound. I hope so for the sake of my children and my grandchildren, that's for sure.
Dr. Sharon Evans, thank you so much for talking with us on the People's Pharmacy today.
I really appreciate the opportunity. Thank you very much.
You've been listening to Dr. Sharon Evans, Professor of Oncology in the Immunology Department at Roswell Park Comprehensive Cancer Center in Buffalo, New York. Roswell Park was one of the first comprehensive cancer centers designated by the National Cancer Institute in 1973. Dr. Evans is a past president of the International Society for Thermal Medicine, which earlier this year honored her with the J. Eugene Robinson Award. We turn now to Dr. Philip Iancu. He is an associate professor in the Department of Investigational Cancer Therapeutics. That's in the Division of Cancer Medicine at the University of Texas MD Anderson Cancer Center in Houston, Texas. Welcome to the People's Pharmacy, Dr. Philippe Iancu.
Thank you so much for having me.
Dr. Iancu, using bacteria as a weapon against cancer seems like this amazing new advance, but in reality, it goes back over 100 years to Dr. William Coley. He was an oncologist, a cancer surgeon in New York City, and he treated quite a large number of patients with sarcoma, very hard to treat cancer, with a surprising degree of success. Perhaps you can tell us how we moved from, something that was lost in the mists of time to your research today.
You are actually exactly right. I mean, it's an idea which sounds very novel and very out of the box, and in many ways it is. But it's actually an idea which is very old. And I mean, Dr. William Coley is the one who needs to get credit for. And he actually used the so-called Coley toxin, which was essentially the kind of a bacteria extract to treat cancer more than 100 years ago. I think the problem why ultimately this method didn't get more momentum beyond Dr. Coley's experiments was that it was also a method which actually had some side effects and some clear disadvantages, which at that time was perhaps relatively hard to control for. And it's an idea which can actually work. I mean, the bacteria can have several effects on the tumor. I mean, one which is relatively obvious is potential direct destruction by the enzymes produced by the bacteria, which can actually directly cause disruption and destruction of the tumor cells. But also, and I would say equally importantly, the bacteria can activate the immune system and can actually also increase the capability of the immune system to recognize cancer as something which needs to be attacked.
Dr. Iancu, you and your colleagues recently presented the results of research that you have done utilizing bacterial spores against solid tumors. So after many years of Dr. Coley's research having been ignored and overlooked, you've resurrected the general idea, but you've really refined the approach. Could you tell us more about your research, please?
We use bacteria, which is specific, I wouldn't say engineered, but attenuated strain of bacteria, which is called Clostridium novii, which is just kind of a normal bacteria which lives in the environment, which is very low on oxygen levels. And that idea actually originated in the lab of Dr. Bert Vogelstein, who is a cancer scientist at Johns Hopkins in Baltimore. And the reason why Dr. Vogelstein's team focused on this bacteria was that they wanted to capitalize on the fact that Clostridium is only capable to proliferate from spores to live bacteria in the environment, which is very low on oxygen levels. And the assumption was that such low oxygen levels are only present in the cancer, but not actually in healthy tissues. So the idea behind that was that Clostridium can be used as a very selective tool, which will do what it's supposed to do in the cancer, but will not affect the healthy tissues. Our study was designed based on the data from dogs which had spontaneous cancers and who were treated with intratumor injection of Clostridium. And it was actually observed that the intratumor injection of Clostridium can actually cause quite substantial regression of the cancer lesions.
Now, you've actually done some clinical trials. That is to say, you have tested this approach in humans. What did you find?
So we actually treated 24 patients with diverse advanced cancers with limited or no therapeutic options. And we tested them with different doses or different amount of Clostridium spores, which were given directly to the tumor. And we've actually noticed several interesting observations. So as expected, we've seen the germination of Clostridium spores, which actually means that the Clostridium spores proliferated in the full-blown organism bacteria, which directly caused destruction of the tumor cells, which was often associated with fever and pain and other symptoms. We've seen that in a little bit less than half of the patients. And these patients obviously had quite substantial changes in the tumor lesions which got injected, which often happened relatively quickly. I mean, we've seen the effect on the injected lesion often within 24 hours, within first 24, sometimes 48 or 72 hours. So we also actually monitored what happens to the lesions which we didn't inject, because the anticipation was that by inducing of anti-cancer immunity, that it might be a mechanism which will help to control not only the injected lesion, but also the lesions which actually were not injected. I should also emphasize that we gave our patients only one single injection, but we have actually seen several interesting outcomes in several cancers, which often were cancers which are known to be hard to treat, such as sarcomas, for which there is relatively few approved therapies with relatively limited efficacy. We've seen that we observed both quite dramatic responses in the injected lesions as well as stabilization of lesions which were not injected.
Dr. Iancu, thank you so much for talking with us on the People's Pharmacy today.
Thank you so much for having me, and thank you so much for giving me the opportunity to share our exciting research experience.
You've been listening to Dr. Philip Iancu. He is an associate professor in the Department of Investigational Cancer Therapeutics. That's in the Division of Cancer Medicine at the University of Texas MD Anderson Cancer Center in Houston, Texas.
We spoke earlier with Don McAdam. His book is The Reinvention of Coley's Toxin. our podcast contains more of our conversation with Don McAdam and an interview with a researcher, Dr. Philip Yanku, about his unique immunotherapy.
Lynn Siegel produced today's show. Al Wadarsky engineered. Dave Graydon edits our interviews.
The People's Pharmacy is produced at the studios of North Carolina Public Radio, WUNC.
The People's Pharmacy theme music is by B.J. Liederman.
To buy a CD of today's show or any other People's Pharmacy broadcast, you can call 800-732-2334. Today's show is 1151. The number again, 800-732-2334, online at peoplespharmacy.com.
When you visit our site, you can share your thoughts about today's show. Have you received immunotherapy for cancer? If so, we'd very much like to hear about your experience.
At peoplespharmacy.com, you can sign up for our free online newsletter or subscribe to the free podcast of the show. When you sign up for the newsletter, you'll get our free e-guide to our favorite home remedies guide. In Durham, North Carolina, I'm Joe Graydon.
And I'm Terry Graydon. Thanks for listening. Please join us again next week.
We hope you enjoyed this podcast. If so, please consider taking a minute to write a review on iTunes. And thanks for listening to The People's Pharmacy.